Okay, I'll turn off my camera for a while just to save the internet band. And let's start. So, hello everyone. I hope you're all safe and healthy. I would like to begin by saying thank you to the organizers for inviting me to participate in this symposium. It is nice to be here with all of you. And today I will talk about spider taxonomy and biogeography. I think I wouldn't have any trouble convincing you of the importance. Okay, just a moment, it should move. Okay. So I wouldn't have any trouble convincing you of the importance of species distribution knowledge for disciplines like biogeography, macroecology, or conservation biology. This is very well illustrated by this map. These are the Conservation International's biodiversity hotspots, which are areas in the world that hold high species richness, high species endemism, and also high extinction threat. Initiatives like these are important to guide conservation decisions and are supposed to represent the world biodiversity as a whole. However, the hotspots are actually delimited based on distribution records of terrestrial vertebrates and angiosperm plants, and nothing on invertebrate animals. It's commonplace to use vertebrates in geographic scale studies because they are supposedly better known in taxonomy, geographic distribution, and natural history. And invertebrates are usually disregarded because, well, they are just insufficiently known. I must confess I don't buy this idea, and I want to illustrate my point with spiders. As you know, we spider taxonomists are a fortunate kind. We have a long cataloging tradition that culminated in the World Spider Catalog. Through this, anyone can access basic taxonomic information and literature on any spider species. We can also get information on the spider species distribution, but only in a very superficial way. We can also count on other initiatives like the Spiders of Europe portal, in which we have not only taxonomic cataloging, but also images and a bit more detailed distribution information. In that case, as far as I know, you can have country records for each species, but nothing more detailed than that. We are here specifically talking about Europe, one of the better known parts of the world regarding biodiversity. But what about tropical, megadiverse, and undeveloped countries like Brazil? This map was published by the late Herbert Levy in the 1960s. The triangles show collecting sites of an early 19th century German expedition in Brazil. The spiders collected in this expedition were described in 1833. By then, only 19 spider species were known from Brazil, and those triangles represent the first detailed distribution records in the country. Now, look at the map at right, which is based on taxonomic literature data. Each green dot represents a distribution record of at least one spider species in Brazil nowadays. And until yesterday, almost 4,000 species are known from the country. It seems we have made some progress in describing our spider fauna and documenting our geographic distribution. But was it enough to use spiders as biogeographic models? To answer that, we must consider the pervasive effect of sampling bias and variation in study effort. I would like to illustrate that with a specific spider family, the goblin spiders or Oenopidae. What you see here is a species accumulation curve for the family. The first Oenopid species was described in 1835 from Europe. It is this small, cryptic spider, nothing apparently very interesting at first sight. With the combined efforts of taxonomists around the world and through time, we reached a total of 444 Oenopid species by 2007. This figure would suggest the family is not particularly diverse and interesting, right? But then, in 2007, we started the Goblin Spiders PBI, an NSF-funded initiative coordinated by the late Norman Platnick. This project congregated over 40 taxonomists worldwide, myself included, which all dedicated to resolve the Oenopid taxonomy, sharing information and specimens, and using the best of the technology available by then. As a result, we discovered an impressive morphological diversity in the group as illustrated by these photos. And of course, we discovered an impressive taxonomic diversity. So, if we had just 444 species known by 2007, with the PBI, we reached over 1,800 species known today. And though the project is finished, most of us still have undescribed species in our labs. This example makes it obvious the impact of variation in study effort on taxonomic knowledge of a group. But what about the geographic distribution? How much do we know the goblin spiders on the map? Well, I can show you detailed information from the neotropical countries. Each green dot in the map in the middle show a published distribution record of at least one oenopid species. Actually, most dots actually represent a pile of records of several species in the same place. This is clear in the map at the left, in which we superimposed a grid of hexagonal cells over the map and counted the number of species recorded inside each cell. As you see by the color scale, we have a lot of geographic variation in oenopid diversity. The map on the right shows a spatial interpolation of species and record density. So the darker the area, more species and more distribution records can be found there. Notice then that we have something like oenopid hotspots of biodiversity. And to illustrate the importance of the PBI, let's look at Ecuador. Most of the species known from the country were described after 2010 by members of the PBI. Actually, most species were first found in the field in a field expedition we held there in 2009. So no one cannot exaggerate the importance of the PBI, but it also makes evidence our knowledge deficits in the group, which can be illustrated in the map as a great gray zone of oenopid diversity. Take, for instance, the place represented by the blue dot. I have no reasons to suppose there are no oenopids there, but if I care to collect them, would I find undescribed species or new records of already known species? Maybe both? We just don't know. And this leads us to preliminary conclusions. So, all we currently know about spider species distribution in the Neotropic is that we don't know enough. So we should concentrate our efforts on collecting and describing spider species and let biogeography to the better known vertebrates, right? Well, I don't think so. Of course, we must increase our sampling taxonomic study efforts, but our ignorance on spider species distribution does not mean the group is useless for geographic scale studies. Most importantly, we must ask ourselves, is the situation that bad? We do know something about spider species distribution, but where is all that knowledge? The answer is it is dispersed through thousands and thousands of taxonomic publications. So all we should do is to assemble all that knowledge. Now let me tell you another story. Back in the late 1990s and early in this century, we had in Brazil an initiative called Biota FAPESP, which funded extensive biodiversity inventories in Brazil. At the time, my PhD advisor, Antonio Briskovit, I suppose most of you know him, coordinated a project within that initiative in which he sampled dozens of localities throughout Brazil. But Antonio also had a simple but also a great idea. He started an effort to compile all the spider distribution records from Brazil available in the taxonomic literature in a single Excel database. And this database made possible for this guy, Ubirajara Oliveira, one of my students, to evaluate in his master's dissertation how much, after all, do we really know about the spider diversity and distribution in Brazil? So this is a publication derived from his dissertation. The map on the left shows the variation in spider species richness in Brazil, and at the right you see the variation in the number of distribution records, all within square small grid cells. Although we still have a lot of sampling gaps, all the white parts in the map, it is possible to make interesting inferences for this data. For example, if we see the relation between the number of species and the number of records within each grid cell, We can identify areas of potential high species diversity and there, like this one in Amazon, and are areas of potentially low species diversity like this one. But most importantly, we can access areas in need of intensive future sampling. But there is more we can extract from this type of data. In this map, we see spider areas of endemism in Brazil, all inferred through a method developed by Ubirajara himself. I would like to compare here our results for two large humid forest biomes we have in Brazil, the Atlantic rainforest and the Amazon. The Atlantic forest is definitely by far the better known Brazilian biome regarding spider diversity and distribution, as illustrated by the density of records we had seen in the maps earlier on previous slides, and also through these areas of endemism. Although there is still a lot of undescribed species there, we are confident that our current knowledge on spiders can be useful to understand the biogeography of this biome. For the Amazon, on the other hand, what we have here are places with high concentration of species in records and shown here, high species endemism. What these areas actually represent are places in which people collected a lot and a lot of specimens through time. Those specimens were made available through collections for taxonomists to study and describe species. So these spots in the Amazon actually represent what I call sampling hotspots, not exactly biodiversity hotspots. And what this map shows us is that what we really need in the Amazon, in other biomes in Brazil, is to increase sampling in the field. But this data can help guide us to find the better places, the most promising places, and the places more in need of field sampling in the future. So, I have been talking a lot about sampling sufficiency, but I should also mention sampling bias. In this slide, you see a bunch of distribution records, and there seems to be some spatial variation in the record density, right? Which can be easily described as a curve. The reason of this variation in record density becomes evident when you note the presence of a road right here. The concentration of distribution records near access routes is among the better known kinds of sampling bias, and it can easily be quantitatively described like this. This is what Ubirajara did in his PhD thesis, but this time integrating spiders to a wider multi-taxon database. So just to show one of his most interesting results. What you see here, you see curves describing the variation in record density in relation to access routes. So for each graph, we have an access route here at left, and the dark curves represent simulations in which we predict how the density records should be distributed in space if the sampling has been done at random through time. And the colored curves represent the real data. So horizontally, you see graphs representing the different biomes or ecosystems in Brazil according to this color scale. And vertically, we show different taxonomic partitions of the data. So what we can see here, we can see that the data are equally biased towards access routes, regardless of the biome or ecosystems, and most importantly, regardless of taxonomic partition. So we see the same problem, the same sampling bias for all the groups together, for the angiosperm plants, for the invertebrates, spiders included, and look at that, also for the vertebrate animals. So these so-called better known taxonomic groups are not that better known, and using them as biogeographic models is as challenging as it is for invertebrates or plants. So now we can reach more sophisticated conclusions. At least part of our current knowledge on species distribution can be used in geographic scale studies. But first, we must compile all the information dispersed in the literature and in museum collections. In other words, first we should know what indeed we know. Only then we can deal with the problems related to sampling insufficiency and sampling bias. And this is true regardless of taxonomic group. Spiders are surely less well known, but not necessarily less useful than other animal taxa for biogeography, conservation biology, macroecology, and so on. And that's it for today. I would like to finish by thanking all my students and collaborators, which you can see here. Everything I have shown here is a product of several years of collecting effort. And of course, I'm also grateful to the several funding agencies that have been supporting our studies. I hope they will continue doing that. And most importantly, thank you all for your attention. And now I can address your questions. Thank you very much.
Thank you, Adalberto. We've actually finished 15 minutes early, so hopefully we've got quite a few questions to get through. But a great talk.
So we have time.
Yes, we've got time. I'm just going to have a look. It looks like we've got one question already. So Alan Cam remarks, great talk, thank you. We see exactly the same type of record distribution in Leicestershire, which is a county here in the UK, approximately 360 species, piles of records from certain locations and lots of under-recorded areas. And then he's provided some links there for your reference if you'd like to have a look.
Nice, thank you very much. Yes, we still need to have in the Neotropics, in Brazil in particular, something you guys have in UK, that all that collective effort, including citizen science, to locate spiders and to locate new distribution records. This is really, really amazing. Well, of course, it's a bit more challenging for us because we're a continental scale country, but it would be really nice to have something like that.
Any more questions? So Martin asks, is there a way of dealing? Oh, sorry, Richard.
Oh, sorry, I was going to ask an actual physical question rather than typing it.
Go for it.
Okay. Yeah, so I suppose the picture in my background highlights a problem because I'm in North Wales in UK and we've got quite a varied topography. So the mountains behind me, I've been attempting to sample spiders on the summits, but obviously it's quite a task going up to the top of these mountains rather than staying down in the valley next to the road. Far easier to sample. So have you got any thoughts about how to encourage people to go off the road to more difficult habitats to maybe fill in those gaps?
Well, this is really, really hard. And I think harder in a country like Brazil. You imagine the Amazon, it's not hard to find images about that in the internet. And you have this vastness of forest. And actually you have a lot of small rivers that can help you find places to collect. So I think the mere fact that we report this problem of records concentrated near access routes can encourage people to go a bit beyond those places. And we have seen some efforts here in Brazil. So the most important is to people to become aware of the problem. And of course, we need to find the resources, the money to do that, because it can be hard to find the most distant places. And you talk about the summits. And I think most of the challenge for the future will be to sample places that are too far, too high, too hard to reach, and also too dangerous. For us in Brazil, it's a bit of a problem, not that big. Of course, you have places in the world in which we have warfare problems and which prevent biological sampling. They have more serious problems. So yes, I think we have the people with the disposition, with the will to do that sampling. We just need more money, more support.
Okay, we've got a couple more questions. Martin asks, is there a way of dealing with the bias and analyzing the data anyways?
Yes, you can include the bias as one of cofactor of your analysis. Birajara have done this in most of his papers. You can try to insert the diversity, the distribution records together with any information or sampling sufficiency, sampling effort or bias as a cofactor. So you can try to find a signal like what's the effect of the climate on diversity, but discounting the effect of sampling bias and sampling insufficiency. Some people also record using, for example, niche modeling approaches, so we can try to expand the node distribution of the species. So for some analysis, at least some kinds of analysis, you can use the polygon, the distribution polygon we infer from ecological niche modeling instead of using actual real distribution records. This can be promising, although you have to deal with all the problems of sampling insufficiency to do the ecological niche modeling. So just like in molecular phylogenetic analysis, there is no single answer, but it is possible to deal with gaps and knowledge insufficiency. But you have to study
Excellent,
a lot.
thank you. And Vicky Wilson asks, how can we find out if your area has already been covered by field studies and access this data?
Well, the most crude and rough way of doing this is exactly looking for distribution records. In the literature, we use, of course, distribution records of described species, but we can also use data from collections. Scientific collections have a lot of material, at least part of it not identified to species, but you can use the simple fact that we have spiders collected in a place, even though not identified, as an indication that there has been some collecting effort in that place, and try to use this information. In the case of our studies with a big database, spiders and other animals and plants, we try to use all the distribution records for each place as an indication of the sampling effort in that place. Maybe not so much for spiders or for millipedes or for plants, but at least that the place has been visited by biologists in the time. So we can compose a kind of index of sampling intensity in one place and use these also as a cofactor of your analysis.
Perfect, thank you very much. And a lot of the other comments here are commenting on the excellency of your talk and how it was a good end to the symposium. So thank you very much for that. We've now got a few minutes. We will probably finish a couple minutes of early, but I'm now going to take this opportunity for some closing remarks. So first of all, I'd like to thank everybody else on the organising committee who were integral to getting this off the ground. That's Dimitri, Jan, Helen Smith, Jeff Oxford, Paul Selden, Richard Gallen and Richard Birkmar. Thank you very much to all of you. Thank you very much to all of our speakers today. I'm sure you'll all agree what an excellent variety of speakers, an excellent variety of topics and an excellent variety of methods where everybody's trying to approach the same sort of goal, which is to advance arachnological research. I think if John, Francis or Mike were here now, I think they'd be sincerely proud of everybody today for their talks, for the outstanding research that continues to advance arachnology, and also to all of you who attended here today for the symposium. The idea of this symposium was to memorialize these great people who each all contributed multiple lifetimes worth of research to arachnology. But especially because we found out about the deaths of two of them, John and Mike, within about a week of each other last year in what I think we can all agree was a terrible year for arachnology more broadly, because we also lost Norm Platnick and Joanne Silwell. It was a terrible year. So this was an opportunity for the British Arachnological Society to commemorate John and Mike and Francis, who passed away in 1995, in a variety of talks, which we've done here today. For the benefit of everyone that's joined us, most, if not all of the speakers today will be contributing papers to a Festschrift. So that's a special memorial issue of arachnology, which is being produced by Paul Selden and Dmitry Luganov, and that will be out early next year. I'd like to thank you all again for your participation, and what we should do now is finish the recording, but if anybody else has any questions that they didn't get in before the talks were over, or you just want to have a quick chat, we'll probably keep the meeting open for another 10-15 minutes or so, just so everyone can catch up. Thank you very much, everybody.